रिकॉर्डिंग हो रही है बेटा?
हाँ जी मैम, हो रही है.
ओके ओके, राइट, ओके, राइट बेटा. So let me continue. I hope that yesterday man finished the structure of male and the female gamete. Is it?
यस मैम, मैम ने हमारे को oogenesis कराई थी कल.
Okay. And also she compared with the spermatogenesis
Spermatogenesis.
and oogenesis. Okay. Yes, beta. Now we have to start with basically two major things are left. One is the fertilization, and after the fertilization, development. And the last major topic that is about that's the menstruation. Okay. So first we'll do that fertilization. Okay. So blackboard is visible to you properly?
Yes मैम. Yes मैम.
Excellent. Yes. So we have to do this fertilization. Okay. So what is this fertilization? As you have been studying in the lower classes, that this is just the fusion of the female gamete and the male gamete. They fuse together and that give rise to the zygote. Right. This is the act of the fertilization. And there is a lot of variation in the fertilization too, but we have to talk about in the humans. So in humans, the fertilization that is internal, okay. So internally the fertilization takes place, and in 10th class also you have studied that in the oviducts the fertilization takes place. So we have to review that just a little bit in more detail, that how the fertilization takes place. So in the fertilization process, sperm's efforts are also there. Role of the uterus is also there. Uterus is also playing its role, and the sperm is responsible for a basic reaction, that is the acrosome reaction. Acrosome is a part of the sperm, you know that, okay? So first, let me see that what is the sperm effort. Sperm effort, the sperm is doing the effort to reach to the site of the fertilization. What is the site of the fertilization? That is the oviducts, okay? The oviducts of fertilization takes place. Ampulla isthmus junction, at that the fertilization takes place. So sperm is doing the effort because sperm, that is a motile structure. So motile, this is motile. Motile means that is able to move with the help of the tailed structure. And you have also studied that in the middle portion, it's full of mitochondria, and mitochondria is providing the energy for its movement, right? Followed? And
Yes ma'am.
so with that, the sperms, they are able to do the effort to move. So near about 200 to 300 millions of the sperms are to be laid in 3 ml of semen, right? But only 100 sperms are able to reach up to the site of the fertilization. So it's not a very easy job in the way many of they are not able to reach over here. And uterus is also playing the role. Contractions of the uterus, okay? Uterus is providing the suitable space for that. Internally also, that is moist too. Mucus is also, it's aligned with the mucus also, right? So uterus, the contractions of the uterus, they also help in the upward movement of the sperms, right? So sperms, they are too aspirated because the cervix that opens into the vagina, and through the vagina at the time of the insemination means when the semen is to be deposited inside the vagina, then the sperms, they enter into the uterus. Then from the uterus, with the moments, they'll be moving, they will be reaching up to the oviducts, okay? Then when they will reach up to the oviducts, and there the fertilization takes place. So what happens after that? When they reach over there, sperm is there, egg is there, both the gametes, they are present. And then what type of the changes are there? The sperms, they are also made capable of doing the fertilization. How they are made capable of? Some of the additional things are there which are present on the outer surface of the sperm, and those additional things, they get removed when they are moving through the uterus, okay, and they are made capable, and some proteins, they are exposed on them, and when they reach up to this female gamete, the ovum, then the ovum on the outer surface is also having certain proteins on that. And when those proteins, they come in contact with one another, and that result in the taking place of the next process, and that is the acrosome reaction. Acrosome reaction means when male, female gamete and the male gamete, when they approach, suppose this is, then the upper part of this sperm, this is the upper part of that you have studied. This is just diagrammatically, I'm drawing that, okay? This one, what is this known as? This is known as acrosome, acrosome. And this is acrosome, what is present? Certain enzymes are present, lysins are there, lysins are present, enzymes are present. And those enzymes, when it come in contact with that, then, and outermost surface of the ovum, it's also having the zona pellucida. Okay, so that you know that it's having the membrane. Two membranes are there. One is the zona pellucida, another is the corona. Yes, what is that? Corona.
Corona radiata.
Very good. Corona radiata. Okay. So passing through that corona radiata, that reaches to the zona pellucida. Zona pellucida will be dissolved by the lysins. Okay. Then it will further enter, and the head portion of that will enter over here. The head will enter into that. Okay. And this releasing up of the enzyme from the acrosome, that is known as acrosome reaction. Right. So acrosome reaction takes place. Now I told you that there are near about 200 to 300 million sperms are there, but 100 of may reach up to that, but only one is to enter into the egg. And how this thing is to be checked, how the nature has made check over here, that to check the polyspermy. How polyspermy is checked? What do you mean by polyspermy? Poly means number of that. Spermy means sperms. The number of sperms may not enter into the egg. This is known as to check of the polyspermy. So only one sperm is to enter into that. So there are two things are responsible for that, to check this polyspermy. One thing, that is the depolarization of plasma membrane of ovum, right? Depolarization. When the sperm come in contact with the ovum, then polarity, certain electrical changes takes place. Depolarization takes place, okay? And that result, we say that depolarization of the membrane has taken place. As a result of that, then second thing, what happened, that cortical reaction is there. Now, what is cortical reaction? Cortical reaction is derived, this cortical term is derived from the cortical granules. Cortical granules. Where these granules are present? Yes, ma'am might have told you that. Where these granules are present? Yes,
Inside secondary oocyte.
this is just beneath the plasma membrane of the ovum, right? So cortical granules, they do have certain chemicals in them. So when the sperm enters into the ovum, then these cortical granules, they burst, right? They burst. They will release the enzymes, right? When these chemicals or the enzymes, I must say chemicals, are released, these chemicals will result in the hardening up of the membrane of the ovum. Okay, what membrane? Zona pellucida. So zona pellucida, that will be hardened up, hard ho jayegi, so that no further sperm can enter into the ovum. So in this way, Entry of only one sperm will take place, not more than one sperm that will take place. So are these two things clear to you? Depolarization of the plasma membrane and the cortical reaction? Yes?
Yes, ma'am.
Okay, good. Now next thing is about that when the fertilization takes place, then in detail we'll see that what happens after the fertilization, but what is the outcome of this fertilization? What is the significance of the fertilization? Already I told you that the ovum is released in the meiosis 2 stage. And at what stage that is arrested? Yes. Can you tell me? Yes.
Metaphase 2.
Metaphase 2 stage, right? This meiosis 2 is there. The primary follicles, they do have the primary oocyte. Then after that, the primary oocyte will be getting converted into the secondary oocyte. And the primary follicles will be converted into secondary follicle, tertiary follicles, and then ultimately that will be getting converted into mature Graafian follicles. And these mature Graafian follicles, they will rupture and they will release the ovum. And that ovum will be in which stage? That will be in the stage of metaphase 2. When the fertilization takes place, then completion of this is the first significance of that. Completion of division takes place. And as a result of that, the ooted is formed from the secondary oocyte, and plus second polar body will be formed. Right? So first polar body already that was formed after the division of the meiosis 1, and that polar body, we are not exactly knowing that what happens to that, but some of they are of the view that first polar body that gets converted into that divides and gets converted into two bodies. So it means that actually one ooted, one ovum, and the two polar bodies or three polar bodies, they will be formed. Clear? Have you followed? So this is the first thing, the significance of fertilization, completion of the division will be there. And this will also result in activation of egg that will be getting activated for further division, right? Now egg is getting, actually this egg is now known as the zygote when the sperm enters into that. So zygote, so activation of that will be there, further division that will takes place, right? And also this restores the diploid number of chromosomes. That's most important, okay? Because gametes are always haploid, half in number of the chromosome, 23 chromosomes are there. So these 23 and 23, they'll make up the diploid number, and the 46 chromosomes are maintained, okay? So this is specific to, the diploid number is specific to any of the species, and always those who are going for the sexual reproduction, gametes are haploid and diploidy is maintained after fertilization, okay? Right? So in this way, these are the some of the important, sorry, one more we can add up, that is the sex determination too. Sex determination, this is the next point of significance. So fertilization result in activation, result in maintaining diploid number, result in the determination of the sex, whether the individual born will be female or the male, right? And just in short, I'll recall back what you have done in the 10th also, that always the ovum, they are haploid, and what type of the sex chromosomes are present in them? One pair of the sex chromosome, that is, sorry, one pair nahi hai, only one number of the sex chromosome, that is X chromosome, okay? The sperms may be of two types, half of the sperm or many of the sperm, they can have the X chromosome, and the other they may have the Y chromosome, because X and Y These are the two sex chromosomes of the males, and X is the sex chromosome of the female. Now it depends upon that which sperm is going to fertilize the ovum. Ovum are all same because they will have only the X chromosome in them. If the sperm carrying this X chromosome fuses with this, that will result in X and X. X and X means one pair of the sex chromosome. The females, they too have that. The child born, that will be girl, right? That will be girl. And if the sperm carrying the X chromosome, that is fertilized, that is fertilizing the ovum, then it will lead to XY combination. This XY combination, that's of the male, and the male child that will be born. Okay, so from here it is also clear that always the sperms are responsible for the sex, not the eggs are responsible for the sex. Eggs, or means I can say that the females are not responsible for determination of the sex, because the females will be producing the same type of the ova, but the males, male means father, the father is responsible for the sex, because that will be producing the two type of the sperm carrying X chromosome and the Y chromosome. Clear? So this is to layman's, this is some of the people, those who are not well educated, they believe that the ladies or the females are responsible for the sex, that that's responsible for giving birth to the girls only. And this is altogether wrong, because the father is always responsible for giving sex of the child. Yeah? So this is all about the fertilization and the significance of the fertilization. Now the next we have to do the development. The development takes place with the in the diploid structure, which is the zygote is formed, and within that zygote, then the further divisions takes place. And those divisions are mitotic division. Okay? And with a little bit different mitotic division. And we name out that procedure or the method, that is cleavage. Cleavage. Okay? Cleavage takes place. And what are the main, you can say that, what are the main characteristics of the cleavage or the special type of the mitotic divisions which takes place in the zygote for the further development, that the cleavage, in the cleavage, interphase is small. Okay? It means it's not of a long duration, quick succession, small duration. And these division takes place in quick succession, right? So बहुत ही fast होती है, and size of the cells which are formed, those cells are known as blastomeres. The blastomeres, they are not of the same size, but they are small size. ठीक है? जैसे एक सेल में दो बने, वो दो क्या होंगे? थोड़े से छोटे होंगे. उस वही सेम साइज के नहीं बनेंगे, right? The size will not be same, a little bit small size. So blastomeres become small size. This is another characteristic feature of that. So shape of that developing thing doesn't change, but within that the number of cells, they increase. When the number of cells increase, the shape should also change, but here the shape doesn't change. Why? To up to certain division, the shape will not change, because the blastomeres, they become small size. Okay? So that will remain the same. These are the characteristic features of the cleavage. Okay. Now let me see that how the development takes place, what further changes are taking place in the zygote. To start with that zygote, first we'll see that what is in short, that what is the structure of this. What is this? This is the egg, right? This egg is bounded by which layer? Two layers are there. One is zona pellucida and another is corona radiata. This is zona pellucida I've shown, and then so this is just diagrammatically showing that so such type of the radiating out cells are there. These things processes cells in the form of, so this is corona radiata. This is zona pellucida, right? Zona pellucida. And what is this? This is the egg. This is egg, right? Now here, the sperm when approaches it, so in the same, I will show that when the sperm, so this is the head of the sperm, the tail one. So that approaches, it's just near. It has penetrated into the corona radiata. Then after penetrating through that, it will reach to the zona pellucida. Okay? So when it reaches this zona pellucida, right, then the license will be released out, then ultimately dissolving dissolving the plasma membrane to the head portion that will enter into that. Okay, the tail will not, the rest of that will not enter into that, only the head will enter into this. Okay, so when it enters into this, then fusion of this genetic material and this, that will take place, and it will result in the formation of zygote. So now this zygote will have the zona pellucida around it. It will have this, okay? So it is not dissolved. From a small region, it will be dissolved and the head will enter, but the rest of that is intact. So it will be intact. So inside that, so this is division, fusion of that. So what is this? This is, now we name out that this is a zygote. Zygote is formed, okay? So that is the zygote, and this zygote will have, this is the diploid nucleus now, okay? This is the diploid nucleus. Then further changes, divisions takes place in the zygote, right? And near about 30 hours, two cells will be formed within that. So we can show the two cells. This is zona pellucida, right? And two cells will be formed. Then after 60 hours, the three cells will be formed. So first two cells. So this is one cell and this is another cell, right? So what is this? This is a two-celled stage. Two-celled stage, right? Two-celled stage is there, and this is about after 30 hours. Then after the 60 hours, okay, 60 hours, the three-celled stage. Three-celled, how you can show that? Should I make the change over here? Because in the book you do have the diagram, okay? So in that, the three-celled stage, the size will not change, but the cell, right there. So what is this? This is now three-celled stage. Three-celled stage, and what's the time duration? That is 60 hours. Okay, 60 hours are there. And then after 72 hours, the four-celled stage is there. Four-celled. So let me go over here. The four-celled stage. Zona pellucida, then corona radiata is no more now. Okay, वो नहीं है. That disintegrates, and four cells will be formed. So in this way, this is four-celled stage. Four-celled stage, right? Four-celled, and that is duration, time duration 72 hours. After that, again, so what are these cells are known as? These are known as blastomeres. So four blastomeres are there. Then this four cell that will be getting converted into 8-celled stage. Then it will be getting converted into 16 to 32 celled stage. So all these four cells, they will be dividing into, each of that will be dividing into two, and total four cells will be getting converted into 8 cells. Okay. So 4 cells will be above and the 4 will be below that, like this. So 8 celled stage. This is 8 celled stage, right? This 8 celled stage is known as morula. This is known as morula. Okay, young morula ye. Then it's getting converted into, further, it's getting converted into 16. 16, then further, ultimately, that will be getting converted into 32 celled stage. 16 to 32 celled stage. Okay? 30, and in that way, group of cells that will be formed. Okay? So group of cells, a mass of cells will be formed in this way. This is mass of cells is there, and this mass of cell, that's known as morula. Morula means mulberry. It look like mulberry. This is known as morula stage. Okay? Morula stage, and the further, just we can say that, uh, don't forget to draw the zona pellucida, forget to draw this. So you have to draw it to zona pellucida too. Okay? Then further, these cells, they get arranged like group of, then there is change in the size of the cells. The outer cells will be small and the inner cells that are slightly big in size, like this. Okay. So what is it? This is also morula, right? This is also morula, large cell, and then ultimately this morula will be getting converted into, again, what is this? This is zona pellucida. Okay. So this is zona pellucida. This is morula. These are smaller cells, outer peripheral small cells, and these are inner big cells, right? Then it's getting converted into, right? It will be converted into big site vesicle, and that vesicle will have a cavity inside that, and it's getting converted into blastula. Okay, should I rub that, this one? Can I rub it up? Yeah.
Yes, ma'am.
Okay. So then it gets converted into blastula. And this blastula will be, this morula will be formed near about 5 to 6 days tak, okay? 5 to 6 day duration mein. And at the 7th day, the blastula, so 7-day duration, the blastula will be formed. So blastula won't be having now the zona pellucida. The zona pellucida that will be dissolved, and it will absorb the, by the time this is also in the uterus, and it will absorb the secretion, become big in size, okay? And a vesicle will be formed. This is big size, and this is the outer cells. These are the outer cells. This is the layer of cells known as trophoblast cells. Nourishing, their function is nourishment. Trophoblast cells, right? And inner cells, here is a group of cells. These are the stem cells. Stem cell means that these are undifferentiated cells. And this is known as embryonic node. That is the embryonic node, right? And this is trophoblast. Okay, trophoblast and embryonic node, and the cells which are in touch with this embryonic node, these are known as cells of Robber. These are cells of robber, right? So what is this structure, whole of that? This is known as morula gets converted into blastula. This is blastula stage. That is blastula stage, okay? So I hope up to this it is clear. Generally in the exam it is asked that draw the diagrams of the development of the zygote up to morula or up to blastula. Okay? This cavity is known as blastocele. Blastocele, right? Blastocele cavity is there. Embryonic. Now, what is the further fate of that? Ab further kya hoga with the blastula? This, I hope blastula is clear to you. Is that clear?
Yes, ma'am.
Yeah. So blastula, further changes are there, that from the embryonic knob, the embryonic membranes which cover the fetus when it is developing inside the uterus, that will be formed. And from the embryonic knob, the cells which are forming this embryonic knob, they will be they will get arranged into 3 layers, and those are known as the germinal layers. You know that the human beings are yes,
Triploblast.
triploblastic. Okay, ectoderm, mesoderm, endoderm is there. That ectoderm, mesoderm, endoderm is formed from the embryonic knob. Okay, then further ectoderm will be given rise to number of organs. The outermost covering of all the organs will be formed from the ectoderm, and the mesoderm, the muscles will be formed, the heart will be formed from the mesoderm. Okay, and the kidneys, gonads, blood vessels, the heart, and the lymph vessels, they will be formed from the mesoderm. From the ectoderm, the ear will be formed, eye will be formed, and the outermost layer of all the organs, that will be formed. And then from the endoderm, lining of the gut will be there, and almost all the glands will be formed. Start like intestinal gland, the gastric glands, endocrine gland, mostly they are formed from the endodermis. Okay, so further this embryonic knob will get arranged into a disc-like structure that will be known as embryonic disc, and the 3 germinal layers will be formed. So, and those germinal layers, when they'll be formed, this trophoblast layer will internally be lined with the endodermis. First of all, endodermis is formed from this embryonic knob, and those cells, they will start getting inside, covering, start covering the trophoblast, right? And it means that now this blastocele will be lined by endodermis. Then that stage is known as gastrula stage. Okay, that will be, and the cavity will be known as gastrocele. Gastrocele. Then further from this, the rest of the endoderm, I told you that endodermis, ectodermis, and the mesoderm, they are responsible for forming all the different organs. So ultimately, the whole of the fetus will be formed from that, and the embryonic layers will be formed from that. Okay. Now, what is the structure of that? It's given in your book too. Let I share that with you. First, let me know that is the things clear up to this formation of the blastula and the gastrula?
Yes, ma'am.
Clear? How the blastula is converted into gastrula? Who will tell me?
Yes, ma'am.
Yes. Embryonic knob will form the different germinal layers. Okay? And endodermis will line the trophoblast. Then that stage is known as gastrula. What is that known as?